Hello everyone and welcome to this cool-ed tutorial video. Today I'm going to be explaining why the 8-channel P800 series is ideal for live cell imaging, and I'm also going to help you to decide which of the two models, the P800 or the P800 Fura, are best suited to your requirements. They do look quite similar and they do share common features, but let's first focus on the differences. The P800 is designed for researchers involved in general live cell imaging applications, with LEDs ranging from 365 nanometers to 740 nanometers, which means that it covers the near IR and can be used with dyes such as size 7. You also have the option to choose between two UV LEDs, either 365 or 400 nanometers, allowing you to pick the optimum UV wavelength for your needs and filter sets. Following the success of the P800, we decided to offer this technology to the calcium imaging community too, and came up with the P800 Fura. The P800 Fura offers supercharged calcium imaging, and instead of the 740 nanometer LED and also the 365 or 400 nanometer LED of the P800, it features a 340 and 380 nanometer LED for ratiometric measurements with Fura 2, and supports this with UV optimized optics. It's also suited to ratiometric fluorescence, pH imaging, FRET, optogenetics, and of course everyday fluorescence applications. The P800 Fura is perfect for labs who carry out a lot of ratiometric calcium imaging, but who also want the flexibility to get involved in other imaging applications as well. And neuroscience labs are one example of this. Let's now look at the common features that make the P800 series ideal for live cell imaging. First up is control. There are lots of different available options which make it very simple to optimise each individual experiment. Although there is no manual control pod, as you can see, the graphical user interface Lightbridge is instead used to control parameters such as irradiance or individual channel switching. Both illumination systems are compatible with a number of third-party imaging software programs, and you can also control them in their compatible USB-controlled TTL trigger box. The P65018 for the P800 and the P65018FR for the P800 Fura. This allows high-speed TTL triggering. We also have a number of control cables, which allows the P800 series to be triggered in different trigger boards, and we also have SMB and BNC cables. Industry-leading sub-7 microsecond TTL triggering is a breakthrough in live cell imaging and ratiometric measurements for capturing dynamic events with the highest temporal resolution and increasing the accuracy of ratiometric measurements. The ability to fit inline single-band excitation filters means expensive external filter wheels are no longer needed, and when used alongside multi-band filters in a pincle setup, offers a low-cost approach to high-speed imaging. This high level of control also makes it simple to protect samples from phototoxicity and photobleaching, for achieving the highest quality results and also pushing the boundaries of time-lapse studies. For example, using Lightbridge for fine irradiance control from 0 up to 2%, and then from 2% up to 100% in 1% increments. This allows you to balance lower radiance while still maintaining image quality. Meanwhile, hardware synchronization with TTL restricts sample exposure to image acquisition and reduces illumination overhead, which is where the sample is exposed to light but the camera is not acquiring an image. Both the P800 and P800 viewer feature the sequence runner function. Controlled through Lightbridge or some imaging software such as Nikon NIS Elements, illumination sequences are triggered with just a single TTL output of a camera or external hardware. This is ideal for ratiometric imaging, and when combined with inline filters, transforms a manual microscope into an affordable and powerful 8-channel automated imaging system. In summary, the P800 is ideal for general live cell imaging applications, whereas if a lab is predominantly using calcium imaging, but there's also a requirement for a little bit more flexibility, then we would recommend the P800 viewer. We hope this tutorial video has helped you understand the P800 series in a little bit more depth, and if you have any questions, please just get in touch. Thank you for watching.